The goal of all diabetes therapeutics is actually to try to reduce blood glucose and try to restore some sensitivity to insulin. Now, the major thing that insulin is known to regulate in the liver that controls glucose production is a protein switch called FOXO. And it's known that insulin inhibits the activity of this FOXO protein, and that prevents FOXO from turning on the enzymes that regulate blood glucose. What's not as well understood is how the fasting hormone actually signals to the liver cells to start making blood glucose. And it's in fact in that area that these new findings sit. A key breakthrough that really led to most of this work was when my graduate student, Maria Mihailova, made the discovery that in fact, looking at where the HDAC enzymes were in the cell, that when she treated with the fasting hormone, that the HDAC enzymes rapidly moved into the nucleus of the cell. So that suggested that, in fact, in response to this fasting hormone, the HDACs were probably regulating the activity of some genes, and we had no idea what, and this was completely unexpected. So then, collaborating with Ron Evans' lab here at the Salk, we set out to decode what was being regulated. And to our surprise, it actually led back to those same enzymes that actually create glucose from scratch inside the liver. Then, in collaboration with our colleagues in Mark Montmany's lab, we showed that these HDACs were directly binding to these glucose synthesis genes and regulating FOXO at these genes. So that means that insulin turns off FOXO function in the fed state, but when we fast, the hormone glucagon is turning on FOXO and glucose production through these HDACs. And in fact, remarkably, lowering the levels of these HDAC enzymes in 4 different mouse models of type 2 diabetes dramatically lowered their blood glucose, cut it more than in half, almost back down to normal levels. So this says that in type 2 diabetic patients where blood glucose levels are sky high, that if we just lower the activity of these new enzymes that we didn't even know were found there in the liver, that this might be a new therapeutic angle. And we're particularly excited about that possibility because, in fact, people working on cancer, and particularly pharmaceutical companies, have been trying to make new cancer drugs and also been working on these HDAC enzymes, thinking that certain forms of cancer could be cured or treated with drugs that inhibit these. So we're excited to see if any of the compounds that have been made by these companies, and maybe not the same ones that will work for cancer, maybe other ones in fact are sitting on the shelves because they weren't useful for cancer, will in fact turn out to inhibit this subset of HDAC enzymes and lead to a new treatment for type 2 diabetes.